Hello, everybody. Welcome to today's lesson. In the last unit, Unit 2, you learned about globular proteins. Do you remember? Yes. Student, today we will start Unit 3, which is about all enzymes, one of the globular proteins. So, in this lesson, we will learn the nature of enzymes and their properties. Now let's start. Enzymes. What are the molecules of enzymes look like? First of all, let's talk about these kinds of enzymes. An enzyme, or all enzymes, are globular proteins with a tertiary structure which gives them a unique three-dimensional shape, a 3D shape. And this shape makes them very unique. Within that very complex structure, all enzymes have one or more pockets or cleft on the surface that is known as active site. The active site of the enzyme is one of the main important characters of the enzyme. The active site of an enzyme is shaped to allow substrates to bind enzymes at these active sites and forming an enzyme substrate complex. Let's look simple representation of the tertiary structure with an alpha helix and a beta pleated sheet. That is the beta chain, and that is the hem. All enzymes are, we said, globular proteins with a tertiary structure which gives them a unique three-dimensional shape. Most enzymes, we said that globular proteins with one or more pockets or cleft on their surface called an active site. If you look at the diagram, the active site is the small cleft which is represented in this simple representation. And the yellow part is the substrate which is attached to the enzyme and makes a substrate complex. Substrate bind to the enzyme at the active site, forming an enzyme substrate complex. Now let's look at a typical example of an enzymatic action, which is the hydrolysis of sucrose into glucose and fructose. If you look, the active site of the enzyme in which the substrate is attached to and binds to make a substrate, enzyme substrate complex. And that complex is the association of the substrate with the enzyme in that small cleft. Now let's look the properties of enzymes. What are the properties of enzymes? First of all, they are all proteins, meaning that all enzymes are composed of proteins, and they are biological catalysts. That means they speed up or fasten a reaction without being consumed in the reaction or used up in the reaction. so they can be used over and over again. Catalysts speed up the rate of the reaction, and remains unchanged at the end of the reaction. That is to mean that enzymes do not incorporate in the product, okay? They are always released after completing the reaction. Enzymes speed up the chemical reaction by lowering the activation energy required to bring about the reaction. The activation energy is the energy which is required to bring about chemical reaction. And the small amount of enzyme can bring about a change in a large amount of its substrate. That means the amount of enzymes, even if small, they can able to fasten a large chemical reactions, and a large amount of product will be produced. Enzymes are very sensitive to pH and temperature. Therefore, they are affected by extreme temperature and a fluctuation of the pH. They can be destroyed by excessive heat. Enzymes cannot tolerate extreme temperature. They are also affected by the concentration of the substrate and the presence of certain substances that act as inhibitor. We said that enzymes are extremely sensitive to pH and temperature. They can be destroyed by excessive temperature. Therefore, they don't tolerate exaggerated temperature or very high temperature. They are also affected by concentration of their substrate, meaning that when the substrate is increased, the reaction will also increases. But when the substrate concentration is lower, the rate of the reaction will be also lowered. And there are also some substances which are known as inhibitors. They can also affect the property of the enzyme. Enzymes cannot make a reaction occur that would not proceed spontaneously without enzymes, meaning that enzymes always catalyze a reaction which are thermodynamically proceeding without enzyme. That means thermodynamically favorable reactions can be catalyzed by the enzyme. Enzymes do not alter or change the equilibrium of the reversible reaction. Never. Okay? Enzymes increase the rate at which reaction reaches equilibrium. Simply, they increase the rate at which the reaction equilibrium is reaching. Now let's talk about a catalyst. What are catalysts basically? A catalyst is a substance that speeds up reaction. The reaction itself is unaltered, not changed, okay? There is no overall change to the nature of the product, and the energy change that takes place during the reaction, and also the catalyst itself. Remember, always catalysts are substances that fasten the reaction, but the catalyst itself do not be changed. And the overall change, okay, there is no change to the nature of the product and the free energy and the catalyst itself. That means we said that Enzymes are biological catalysts. They don't alter by the reaction, even if they speed up the reaction. Okay? Enzymes allow biochemical reaction inside the cell to take place quickly or fastly at a temperature that will not damage the structure of the cell. Okay? What are enzymes? Why are enzymes specific? Okay? This is also a function of the active site because of the conformation of the active site. That means the way in which it is shaped. That is known as conformation. Only a certain substrate or combination of substrate can bind with it. Therefore, we say that enzymes are specific to a particular or a group of similar substrates. Enzymes typically catalyze only one or a few similar chemical reactions because they are specific in their choice of substrate. Each and every enzyme is specific either to a particular type of substrate or with a few similar kinds of substrate. Okay? This specificity is due to the active site of the enzyme, which is shaped so that only a certain substrate molecule will fit into it. That is what you call the active site, where the substrate is attached. Enzymes are sensitive to their environment. This is simply to mean that the activity of the enzyme is influenced by both temperature and a pH. Most human enzymes, for example, a protein-degrading enzymes like trypsin or pepsin, work best at a temperature about 40 degrees centigrade and within a pH range of 6 to 8. Mostly, the optimum temperature for those enzymes which are said to be found inside the stomach, pepsin and or trypsin, is about 37 degrees centigrade up to 40 degrees centigrade. And the pH, the temperature is about 37 up to 40 degrees, but the pH is ranging from 6 to 8. This is for trypsin. Now let's look how are enzymes affected by temperature. Temperature affects enzyme action in two ways. The first one is a higher temperature gives enzyme molecules and the substrate molecules more kinetic energy, and they move around faster and for more enzyme substrate complex to be produced. That means increasing the temperature will increase the random molecular movement. This increasing the random molecular movement will also provide an opportunity the molecules to collide and change into another new substance that is called a product. A higher temperature affects a chemical bond holding a tertiary structure of the enzyme in place, particularly those in the active site. As more and more of these bonds break, the shape of the active site will be changed, and it can no longer bind with its substrate. That's why we say enzymes are extremely sensitive to higher temperature. Okay? Of course, increasing the temperature, even if it increases the rate of the reaction, but extreme increment in temperature can change the shape of the active site. How enzymes are affected by pH. Now let's look the other factor which is affecting the property of the enzyme. pH affects the enzyme molecule in a similar way to higher temperature. That is to mean that a pH that is too low, that is to mean that too acidic, or too high, that is to mean that too alkaline, will cause changes on the active site to alter, to be changed, and cause the active site to lose its conformation. The substrate cannot bind, and so the reaction is no longer catalyzed because the active site is already changed. In the cell, what will happen? In the cell, some reactions are thermodynamic feasible. That means they can proceed spontaneously, but do not occur at appreciable rate, okay? Therefore, the only reaction that occur at appreciable rates are those from which an enzyme is present. That is to mean that even if there are so many thermodynamically feasible reactions within the cell, the rate is very low. Therefore, the only reaction that occur at appreciable rate or faster rate are those which is catalyzed by the enzyme. Okay. And again, all reactions are mediated by biological catalysts called enzymes. All biological, chemical reactions are facilitated by the enzyme. Now let's look the other important term known as activation energy. What is it? Activation energy is the energy required by the molecular system or the cell to allow a chemical reaction to start. That means the rock on the cliff must be pushed to roll down the hill. Just like that, in order to start a chemical reaction, there must be a starting force, okay, or an energy which initiates the reaction to proceed. The activation energy is a minimum amount of energy needed to start the reaction, leading to the formation of a high energy intermediate. Now, how to overcome the activation energy barrier? We said that the cell, in order to chemical reaction to occur within the cell, okay, there must be an activation energy. Therefore, how the cell overcome the activation energy barrier? How they resolve this problem? First of all, in order to overcome the activation energy barrier, either heat, which is very fatal to the cell, the chemicals or the molecules must have exposed to heat in order to react. But this is fatal to the cell, very dangerous to the cell. Or must lower the activation energy. How? It is by the catalyst. Which catalyst? The biological catalyst. The biological catalyst is the enzyme. Now, students, let us do this activity. Indicate whether the statement written below is true or false. Is false? Change the word or phrase needed to make the statement true. Now let's look the question. Look, the first one. Enzymes are able to reduce activation energy of chemical reaction in the body. That is the first question. You may say true or false. But if you say false, change the word or the phrase needed to make the statement true. Okay, you need to make it true. The second question is, enzymes are able to reduce the activation energy of chemical reaction in the body. The third question, in biological system, a catalyst is called an enzyme, which lowers the activation energy of the system. You have a few minutes to try these, of course, two questions. The first two are very similar. Therefore, the first question and the third question should be attempted to be answered by you. Students, I hope everybody attempt to answer the question. Let's do it together. Enzymes are able to reduce the activation energy of chemical reaction in the body. This statement is true. The second one, enzymes speed up chemical reaction. However, they are consumed by the reaction. That is a false statement, okay? The statement should have to be corrected in this way, okay? Enzymes speed up chemical reaction, however, they are not consumed by the reaction. The third one, in biological system, a catalyst is called an enzyme, which lowers the activation energy of the system. Did you say true? If you say true, that's correct. And finally, this is the end of today's lesson.